Hello, my name is Veronika Magdanz, and I will present to you the development of flexible magnetic 3D printed robots for cell and drug delivery. Soft robotics is an emerging technology that has high potential for applications in biomedicine. The advantages of soft materials are their adaptability, for example, allowing them the motion through tight spaces or changing the shape, and also the gentle interactions with cells and tissue. Also, soft materials are more biocompatible and even biodegradable. They often can also be loaded easy inside the network, and they show many different types of responsiveness to stimuli. Also, the imperceptibility is an interesting advantage that can be helpful for the development of artificial skin. The potential impact for soft robotics in biomedicine is for the development of active scaffolds for the delivery of drugs, cells, or tissue. Also in regenerative therapy, it's very interesting for the development of artificial skin, for stem cell delivery, or for implants. For the development of such soft robots, we are looking at nature and are inspired by the motion of sperm cells, for example, because this flexible flagellated motion is one of the main propulsion mechanisms for motor cells on the microscale. So we are inspired by this type of motion, so we want to develop flexible robots that move in a similar fashion. But first we have to tune the material properties to achieve such flexible materials. So in this proof of concept study, we use PDMS and tune its elastic properties by tuning the ratio of the monomer and the curing agent, for example, from 1:5 to 1:30. And thereby we can increase the flexibility, which is shown here by a decrease in the elastic modulus. Then we create a 3D design of sperm mimicking shape. So we have here this 3D design, which are then we use in 3D printing to create such flexible filaments, which we can see here, and which are then cured at high temperature. And subsequently, we deposit the magnetic material, in this case 100 nanometers of nickel, on top of this structure. We do this magnetic deposition because we want to be able to actuate these flexible filaments in a magnetic field. Here we have on the left our setup of electromagnetic coils, which we are then using to create an oscillating magnetic field. We position our sample in the center of these electromagnetic coils, and then we can see that these filaments are starting to actuate and to move forward under the influence of these magnetic fields. When we track these samples, we can also see the oscillations in this trajectory, and further we can use the magnetic field for the directional control of the robots. When we look at the different factors that influence this flexible motion, we can see that with increasing frequency of the oscillating magnetic field, we increase the velocity of the robots up to about 4 Hertz. Above this frequency, the filaments are not able to follow the magnetic field, and it's called the step-out frequency, and the velocity is reduced. With increasing flexibility of the robot, we can also achieve high velocity of the flexible robots. And finally, the location of the magnetic segment on this flexible filament does not play a major role and does not influence the velocity so much. Next, we explore the potential for cell delivery. So as a proof of concept, we culture skeletal muscle cells on top of these PDMS filaments for several days, and we can see that the cells are healthy and that they can also differentiate into myotubes and muscle fibers on top of this PDMS filament. Next, we are also exploring smart materials. So we go away from the PDMS and go to hydrogels, in this case, thermoresponsive penile compactor filaments. And these have the ability to respond to temperature by a change in their size and in their shape. So we can take advantage of that for not only the shape change, but also for targeted release of cargo. To demonstrate that, we load these filaments with fluorescently labeled mesoporous silica nanoparticles. Then we increase the temperature, which causes a contraction of these hydrogel filaments, which expels water and thereby also expels the the particles inside this hydrogel. So this mechanism can be used for targeted release upon a temperature change. If we embed gold nanorods into the hydrogel, we can also use that to use a photothermal response of the filaments and use light to trigger this release. So in principle, we want to develop cell-laden flexible robots in the future for the delivery and positioning of tissue, cells, and molecular cargo. Thank you very much for your attention.